Hi everyone, today I represent my group members to explain about glycolysis, Krebs cycle, and electron transport chain. The cellular respiration occurs in three stages, which are glycolysis, Krebs cycle, and electron transport. Glycolysis is an anaerobic process, while Krebs cycle and electron transport are aerobic processes. This diagram shows the simple explanation for glycolysis process. For your information, glycolysis is the metabolic pathway that converts glucose into pyruvate and a hydrogen ion. There are three final products of glycolysis, which are two pyruvate molecules, two ATP and two NADH. Glycolysis can occur in aerobic and anaerobic conditions. In the presence of oxygen, glycolysis is the first stage of cellular respiration. In the absence of oxygen, glycolysis allows cells to make small amounts of ATP through a process of fermentation. All of the glycolytic enzymes are found in the cytoplasm. There are two phases in glycolysis, which is phase 1 preparative phase and phase 2 ATP generating phase. Now, let me explain about 10 steps of glycolysis. The first step is the reaction catalyzed by enzyme hexokinase. In this step, the conversion of diphosphate into glucose 6-phosphate. The glucose molecule is phosphorylated, receives a high energy level to become glucose 6-phosphate. The reaction is catalyzed by enzyme hexokinase. The second step is the reaction catalyzed by enzyme phosphoglucose isomerase. In this step, glucose 6-phosphate, a more reactive molecule, is rearranged to become the isomer fructose 6-phosphate by enzyme phosphoglucose isomerase. The third step is the reaction catalyzed by enzyme phosphofructokinase. The fructose 6-phosphate is further activated by the addition of another phosphate group from hydrolysis of ATP by enzyme phosphofructokinase with magnesium as a cofactor. The step 4 is the reaction catalyzed by enzyme aldolase. Enzyme aldolase split the fructose 1,6 biophosphate into glyceraldehyde 3-phosphate and its isomer, dihydroxyacetone phosphate. The step 5 is the reaction catalyzed by triophosphate isomerase. Dihydroxyacetone phosphate is rearranged into glycerate 3-phosphate. The step 6 is the reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase. In this step, glyceraldehyde 3-phosphate dehydrogenase, where NAD+ is reduced to NADH, and inorganic phosphate is attached to the substrate making the product glycerate 1,3-phosphate of very high potential energy. The step 7 is the reaction catalyzed by phosphoglycerate kinase. 1-phosphate from each glycerate 1,3-bisphosphate is transferred to ADP, form ATP and glycerate 3-phosphate by enzyme phosphoglycerate kinase. The step 8 is the reaction catalyzed by phosphoglycerate mutase. In this step, the glycerate 3-phosphate is rearranged to form glycerate 2-phosphate. The step 9 is the reaction catalyzed by enolase. In this step, removal of water molecules from glycerate 2-phosphate by enzyme enolase produces phosphoenolpyruvate. The step 10 is the reaction catalyzed by pyruvate kinase. In this step, the second phosphate is transferred to ADP to form ATP. Phosphoenolpyruvate is converted to pyruvate. In the aerobic respiration, most organisms will undergo two more steps, which are Krebs cycle and electron transport, to produce their ATP when oxygen is present. In the eukaryotes, It is occur in mitochondria. In prokaryotes, it is occur in cytoplasm. This is the simple diagram explanation for Krebs cycle. The Krebs cycle is known as tricarboxylic acid cycle. It involves the oxidative metabolism of acetyl units and serves as the main source of cellular energy. It converts pyruvate to carbon dioxide, NADH, FADH2, and GTP or ATP. The first step of Krebs cycle is the acetyl CoA joins with oxaloacetate to form citrate. The citrate is then converted to isocitrate. Isocitrate is then oxidized to alpha ketoglutarate, which results in the release of carbon dioxide. One NADH molecule is also formed in this step. Here alpha ketoglutarate is oxidized to form a 4-carbon molecule which picks up coenzyme A forming succinyl CoA. A second molecule of NADH is produced alongside a second molecule of carbon dioxide. In step 5, succinyl CoA is then converted to succinate and one GTP molecule is produced. Then, succinate is converted into fumarate and a molecule of FADH2 is produced. In step 7, fumarate is converted to malate. Malate is then converted into oxaloacetate and NADH is also produced here. The electron transport chain. While Krebs cycles occur in the matrix of the mitochondrion, the electron transport system chemicals are embedded in the cristae. In the ETS, NADH and FADH2 are used to produce ATP in the cytochromes. This is called oxidative phosphorylation, as the energy to synthesize ATP is derived from the oxidation of hydrogen carriers. In the first step of electron transport system is generating a proton motif force. NADH and FADH2 transfer the hydrogen atoms to specific carriers on the inner membrane of mitochondrion and are oxidized back to NAD+ and FAD respectively. The hydrogen atoms are passed along carriers and then split into their hydrogen ions and electrons along the pathways. In complex 1, the 2 electrons removed from NADH are passed to flavoprotein, then to coenzyme Q. Complex 2 transfers electrons from succinate to coenzyme Q. Coenzyme Q transfers the electrons to complex 3. Complex 3 accepts the electrons and passes them to cytochrome C. Cytochrome C passes the electrons to complex 5. The step 2 is chemiosmosis and ATP synthase activity. In this step, electrons are passed from one carrier to another. The carrier molecule gaining an electron is reduced, and the carrier molecule losing the electron is oxidized and able to accept more molecules. The last step is oxygen acts as the final electron acceptor. The final electron acceptor at the end of the transport chain is oxygen. Complex 5 transfers the electrons to oxygen, which combines with hydrogen ions to form water. In conclusion, the products of cellular respiration are needed for photosynthesis and vice versa. Together, the two processes store and release energy in virtually all living things. That's all from my teams. Thank you for watching.